Okay, hello everyone. Please, I want you to prepare. Can you hear me first?
Yes.
Prepare paper 1. We'll try to solve as much questions as we can. We will solve questions on biological molecules. Okay. And I want to start solving I want you to start solving what's this called? The recent exams. So we will start from May, June 2020, and we'll try to finish from there. Okay, now I'm not going to wait for anyone. I'm going to start immediately. Let's look at number 6 here in the June 2020 exam. Now, if you noticed that biological molecules is a lesson that most commonly appears in paper 1, more than it does in paper 2, and in every exam, you have at least 6 questions, 5 to 6 questions on biological molecules. Okay, so let's read together. Here, the flow diagram shows the results of a number of tests on a solution of biochemicals. So you have a solution of biochemicals here, and it will have, then we did a Benedict's test, and the result was blue. If the Benedict test was blue, then does it contain reducing sugar or not?
No.
No. Okay, there was a non-reducing sugar test, and it was yellow. So this contains non-reducing sugar, or it doesn't?
It does.
It does, very good, because the starting color is blue, and then it starts changing color, green, yellow, until it is brick red. Now, the iodine test was blue-black, so does it contain starch? Tell me, Abdullah, does it contain starch if the iodine test is blue-black?
Yes.
Yes, it does, very good. And the burette test is purple, so does it contain protein?
Yes.
Yes, so it does not contain reducing sugar, no reducing sugar, but it does contain non-reducing, and it does contain starch, and it does contain protein. Okay, which substances are present in the solution? Who will give me an answer? What is the only non-reducing sugar that we studied? It's very easy to spot the answer, by the way. We studied one example of non-reducing sugar. Which was it?
Glucose.
No, non-reducing. I said non-reducing sugar. Glucose is...
Sucrose.
Yes, so the answer is either C or D. Okay, so this will make us narrow down the answer. Okay, and then we said that iodine is positive, so it contains starch. And look at C. Is C the correct answer or the wrong answer? Did we do any test for fats?
No.
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 So is C right or wrong?
Wrong.
Wrong. So what's the remaining answer? Come on.
D.
D. Okay, the answer is D. Okay. Okay, let's move on. Now, this is so unpractical. This very strange pen. So I'm going to erase that. I have to keep on erasing. This is so bad. Okay, give me a second. Give me a second. So I don't have to keep on erasing. Okay, let me use this. One minute. We were which page? We said we will start from 1/20/20. Tell me which page, girls? 149. Yes, we were here. Okay. So let's go to number 7. Read number 7. Maltose and sucrose. We are on page 149 because we started from 2020 exams in case you just attended. Maltose and sucrose are disaccharides. Maltose is formed from 2 molecules of glucose, while sucrose is formed from 1 fructose and 1 glucose. Which row shows the molecular formula for the 2 disaccharides? Now this is more than easy. Who will give me an answer? I'll choose someone because you're all silent. Yassine, what is the formula for 1 monosaccharide?
A monosaccharide, it's C6H12O6.
C6H12O6. Okay. Now, if we will double that, C6, H12.
It's C12.
Yes.
H24O12.
Okay, O12.
And then we take H2O from it, so it's C12.
Excellent.
H22O11.
Excellent. Now, isn't maltose and sucrose, both of them are disaccharides?
Yes, so it's A.
Yes, excellent, correct. If someone has any objections or don't understand why, just ask. Okay, 8. The diagrams show short sections of some common polysaccharides and modified polysaccharides. So, we have 1 and 2 and 3. 1 is something that we are not familiar with, but I think we are familiar with number 2. What is number 2? Who will tell me?
Cellulose.
Yes, it is cellulose. I think you noticed that the, what's this called? Every other beta glucose molecule is rotated. You see the oxygen here is above, it's high. And number 3, what is number 3? So 2 is cellulose. What is number 3? Who will tell me number 3? It's made up of alpha glucose or beta glucose. 2 alpha glucose molecules.
Made up of 2 alpha glucose.
2 alpha glucose. So what is 3? It's a disaccharide made up of 2 alpha glucose.
Miss?
Yes.
Miss, the sheet screen is lagging.
Is it now? Is it clear now? Can you see it now?
No.
Yes or no?
No.
No? Okay, let me share again. I'll stop sharing and then. Is it obvious now?
Yes.
Okay.
Thank you.
You're welcome. Okay, tell me 3. I have alpha glucose and alpha glucose. This will give me which disaccharide?
Maltose.
Maltose. Very good. So 2 is cellulose because it's a beta glucose and beta glucose. How did I know that it's a beta glucose? Go to carbon number 1. I'll see that the hydrogen is below. So as long as the hydrogen is below the ring at carbon number 1, this is a beta glucose. Okay. And if you look at the second beta glucose, look at the oxygen here. It has been inverted. It was up there and now it moved down to be here. So this is an inverted beta glucose. So number 2 is very clear. Number 2 is a glucose. Number 3 is a maltose. Because this first ring is an alpha glucose. See, the hydrogen at carbon number 1 is above the ring. And the second ring also has the hydrogen at carbon number 1 above the ring. So this is alpha glucose and this is alpha glucose. So number 3 is maltose. Number 1, we did not take it. Okay. The polysaccharides can be described as F is composed of beta glucose monomers with 1 to 4 glycosidic bonds. Where is F? Oh, this is F. There are described. So this is F. G is composed of alpha glucose monomers with 1 to 4 glycosidic bonds. So number 3 is G. H is composed of N-acetyl glucosamine monomers with beta 1 to 4 glycosidic bonds. Of course, this is what we did not take, the strange compound to us. 1 will be H. Do you all agree? So what will be the answer? Which row correctly matches the numbers, the numbered diagrams to the descriptions of the polysaccharides?
B.
B.
Yes, correct, B. Okay, good. Now, 9. Which molecules contain at least 2 double bonds? Let me see. Unsaturated fatty acid, collagen, and hemoglobin. Okay, this also is easy, but it
Does collagen have double bonds?
isn't collagen a protein?
Yes.
And the protein is made up of many, many, many amino acids bonded together. What is the formula for a single amino acid? You have two carbon atoms bonded together, and then you have an amine group, NH2, and then you have a carboxyl group at one end, and the central carbon is bonded to a hydrogen and the variable R group. So you have a double bond here. In every amino acid, you have a double bond. Imagine having so many amino acids bonded together. So
Okay.
collagen has at least two double bonds or not?
Yes.
Yes, okay. And the same case for hemoglobin. Now the unsaturated fatty acid, does it have a double bond or doesn't have a double bond?
It has a double bond.
It has. Very good. So what's the answer?
A.
A, very good. It's easy when you just study well and know exactly the structures of all the biological molecules we studied. Okay, very good. The diagram shows a protein molecule. Two long polypeptide chains form alpha helixes for much of their length, and these twist together into a fiber. At one end, each of these polypeptide coils into a globular head. Two short polypeptides bind to each globular head. Okay, it is describing the structure of this protein molecule. It's a molecule that we did not study. We only studied two. We studied collagen and we studied hemoglobin. So this is something that we did not study, okay? But what describes the protein structure of this molecule? Is it a quaternary structure because each molecule consists of 6 polypeptides? Or is it a secondary structure because the long polypeptides form alpha helix? Or is it a tertiary structure or a tertiary structure? This is made up of more than one polypeptide chain, as you can see, and they are all working together as one unit. Which level of protein is that? Primary or secondary or tertiary or quaternary?
Quaternary.
Quaternary. Okay, without even thinking too much. Now, to cut a long story short so that you're never confused, the primary and the secondary and the tertiary protein structures, all of that, we are still talking about one polypeptide chain. The primary structure, that single chain, is a straight chain made up of a certain sequence of amino acids. The secondary structure, that primary chain will coil or fold. The tertiary structure means that that secondary structure itself will fold some more to form a three-dimensional shape. So level 1 and 2 and 3, we are still talking about one single chain that will keep on folding and folding and folding. Quaternary structure means more than one chain will work together. Maybe they're 2, maybe they're 3, maybe they're 4. So it's definitely A. So you understand the different levels? I hope you have no questions. 11. Which type of bond will keep a folded protein in its precise shape for the longest time as the temperature rises? You have disulfide, hydrogen, hydrophobic, and ionic. These are the 4 bonds that we said determines the shape of the tertiary protein between the R groups. Which bond is the strongest bond, we said?
Disulfide.
Yes, the disulfide is the strongest bond. The weakest is which one?
Hydrogen.
Hydrogen. Okay, up till now, any questions? Did we do any mistakes? Go
No.
check. Are you sure? Okay, very good. Now, June 2020, paper 1-2. A student carried out the Benedict's test on a sample and got a negative result. So as long as she got a negative result, this sample contains reducing sugar or no reducing sugar?
No reducing sugar.
Okay, I want you all to participate, please. No reducing sugar. What should the student do to confirm there are no sugars present in the sample? Read A, B, C, D. I'll give you a second and tell me. So now we want to do the test for the non-reducing sugar. What is the test for the non-reducing sugar? Abdullah. Is Abdullah there? Where is he? Abdullah comes and goes. I don't know where is he. What is the test for non-reducing sugar to make sure that non-reducing sugar is not present? Give me an answer. Serene,
Yes.
can you
This
tell me?
it will be C.
Okay, very good, because the test for the non-reducing sugar is that we will boil with hydrochloric acid. Can you remind me, why do we boil with hydrochloric acid?
To break the bonds.
Very
To
good.
break glycosidic bonds.
To break the glycosidic bond and to break the disaccharide into two monosaccharides. It will provide the water needed for the hydrolysis reaction. And then we will neutralize it first. And then we will repeat with the Benedict in order to make sure that there is no sugar of any sort present. If you have any questions, stop me. Now the diagram shows 3 hexoses. Hexoses means they contain how many carbon atoms per ring?
6.
6, okay. In which carbohydrate do these 3 hexoses occur? Sucrose, this is the easiest question in my opinion. What's the answer? Sucrose is made up of what? Mariam, tell me, sucrose is made up of which 2 monomers?
Alpha glucose and fructose.
Okay, which is fructose? Which shape is fructose? 1 or 2 or 3?
2.
2. So immediately sucrose will contain the fructose here. So the answer is automatically C. If you want to check, cellulose it should contain beta glucose. Is number 3 a beta glucose? Look at carbon number 1.
Yes.
The hydrogen is below the ring, yes. Glycogen is only a polymer made up of what? Made up of alpha glucose. So 1 should be an alpha glucose. Look at 1. Is it an alpha glucose? Go to carbon number 1, hydrogen above the ring. Yes. So the answer is definitely C. No other answer. Okay. Now, the diagram shows part of the chemical structure of a natural occurring polysaccharide. What type of glycosidic bonds are present? Okay, this is a nice question. Do you want me to help you or you will solve it alone? I don't want you to look at the mark scheme. I want you to look at the bonds. Bonds are given names, and these names, they go to the carbon atoms. Let's look at the first ring. You will go look at this oxygen here. This oxygen is upright. So we will move in a clockwise direction, and this will be carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and carbon 6 is up there. Now let's look at the second ring. Again, oxygen is up here, so it is upright. So the number of the carbon atoms will move in a clockwise direction. 1, 2, 3, 4, and 5. So this bond is between carbon what and carbon what?
1 and 3.
1 and 3, okay? So the answer is either A or C. Now the second one, look at the second one. This is carbon 1, and look at this ring. This is carbon number how much? 1, 2, 3, and 4. So the second bond is what and what?
1 and 4.
1 and 4. Actually, A and C are also 1 and 4 and 1 and 4, but the difference is whether these are beta glucose or alpha glucose. They are beta or alpha glucose. Let's go to carbon number 1. The hydrogen is above or the OH is above. The bond is between 1 and 3. So up there, do you see a hydrogen?
No.
No. So this is beta or alpha glucose?
Beta.
Beta. So the answer is what? C. Do you all understand? Do you all know why this is? These are made up of beta glucose molecules. Go to any carbon number 1 and look at carbon number 1. You'll find that up there you don't have a hydrogen. It was an OH. It was originally an OH group. So the hydrogen was below. So this is a beta glucose. Any questions? What is the maximum number of water molecules produced when a triglyceride is synthesized? Now, this is so easy. How many water is made?
3.
Okay, how many are you there? I want all of you please to join, okay? Now, where am I? Where did this go? Do you all agree that in a triglyceride, 3 water molecules are made? Every glycerol binds with a fatty acid, gives water. Okay, Yassine, 11.
Yes.
What occurs during the formation of a peptide bond between 2 amino acids? Read A, B, C, D, and tell me.
B.
Okay, let me read. OH is removed from the carboxyl and hydrogen from the amide. Yes. Now, I told you here a very nice trick to remember. I told you when you come to study a peptide bond, you will draw 2 amino acids, okay, NH2, and you will draw the whole structure of the amino acid. You have a hydrogen here and a variable R group. Next to it, another amino acid, carbon, the amine. I don't have to draw it again, but you have a amine group next to a carboxyl group. I will change color to remind you because we have a peptide bond that forms between 2 amino acids, and then we have hydrogen bonds that come next. The peptide bond is between the hydrogen of the amine and the OH from the carboxyl. This will give us water, and this is a condensation reaction, and we will have a peptide bond here between the nitrogen and the carbon. Now, when this hydrogen bonds with this oxygen, what does this give me? A peptide bond or a hydrogen bond?
Hydrogen.
Hydrogen bond. Okay, this hydrogen bond, why is it important? Because we have different levels of protein. When the amino acids, they make a straight chain, and there's a sequence of amino acids. This is which level?
Primary.
Primary, very good. And then this single chain of amino acids, when it coils up and it is held in place by hydrogen bonds, or it folds and it is also held in place by hydrogen bonds, this is which level? Level?
Second.
Very good. The second level or the secondary protein level. Here, the hydrogen bonds is between the hydrogen of the amine and the oxygen of the carboxyl of the amino acids. Then we have the tertiary structure in which I have this coil or this folded sheet will fold some more to form a precise 3-dimensional shape. What are the bonds that hold the protein in this precise 3-dimensional shape? Who studied? Abdullah, can you tell me? We have 4 other bonds that are responsible for the precise 3-dimensional shape of the tertiary protein. Who will remind me what are those bonds? Yassine.
Yes.
Excuse me? I'm asking a question.
Yes.
What are
What's
the
the question?
bonds? What's the question? What are the bonds that will hold the tertiary structure of the protein in a precise 3-dimensional shape? Mariam, can you help him?
The polypeptide.
The polypeptide? What? No, this is not the name of a bond. We have HHID. What's the first H?
The hydrogen
Hydrogen.
bonds.
Hydrogen. This hydrogen bond is not the same as the hydrogen bond found in the secondary structure. This hydrogen bond in the tertiary structure is between the R groups of the amino acids. And then what's the second H for? Hydrophobic bonds, again between the R groups, between the hydrophobic or the non-polar R groups. I for the ionic bonds between the charged R groups, and the D for the
Sulfide.
very good, disulfide bonds, again between the R groups of the amino acids. In the tertiary structure of the amino acid, the folding here takes place between the R groups or the side chains of the amino acids. Okay, I know you did not study, but do you understand everything I'm saying? Do you recall? Do you remember?
Yes.
Good. The diagram represents roles of water in living organisms. Yassine, this question is for you. The first function is that it carries dissolved substances. The second function, water provides a constant environment for aquatic organisms. The third function of water, temperature control due to evaporation. Which roles are dependent upon hydrogen bonds between the water molecules? 1 or 2 or 3 or which?
Number 2 and number 3.
Yes, you are correct. 2 and 3. Now, can you explain why? The first function, that water carries dissolved substances. It depends on the presence of hydrogen bonds in water or another feature.
Another feature.
What is it?
I think it was the R groups. They depend on the solubility of the.
Yassine, I'm talking about water. I'm not talking about proteins, my dear. There are no R groups in water. Water, H2O. Why is water a solvent and why does it carry things around, and why do a lot of ions and polar molecules, I'm trying to remind you, dissolve in water?
Because they are charged.
Yes, because water is a dipole. It has an electronegative oxygen bonded to two electropositive hydrogen. Okay? What's going on here? One minute. Okay. Now, is this question clear? Yassine, now do you understand this question?
Yes.
Okay. Now, June 2020, variant 1.3. Then we finished this year. The color of a positive Benedict test is due to the formation of copper oxide. The mass of the copper oxide is proportional to the mass of the reducing sugar present. Samples of fruit juice were tested for the presence of reducing sugars and non-reducing sugars using the Benedict. The table shows the mass of copper oxide after boiling with Benedict and after acid hydrolysis and boiling with Benedict solution. Which sample contained the most non-reducing sugar? Okay, this is a tricky question. Can you look at the numbers and tell me which one contains the most non-reducing sugar? Look at the mass of the copper oxide. Look at the mass of the copper oxide. After boiling with the Benedict solution, you have a mass of 20, 30, 50, 65. And then after acid hydrolysis and boiling with Benedict solution. Why do we boil with acid? To find out that we have which type of sugar when we boil with acid?
Non-reducing.
Reduce.
Non-reducing. Okay, now which one has the most mass of non-reducing sugar? And how did you calculate it? Tell me. No answer? They said here that the mass of the copper oxide is proportional to the mass of the sugar. When I boil with Benedict, for example, and then I have 30 milligrams of copper oxide because I boiled with Benedict, and then I'm going to add an acid and heat it and neutralize it and boil with Benedict again. And then I will find a mass of 45, a total mass of 45. 30 milligrams of copper oxide were present at the beginning because of the reducing sugar. And then when I boil again with hydraulic acid and do the Benedict test again, now the total mass is 45. So what was the mass of the non-reducing sugar? Give me an answer. Look at A. When I did the Benedict's test, 20 milligrams of copper oxide appeared. And then when I did the test for the non-reducing sugar, the same mass appeared. So does A contain non-reducing sugar?
No.
No, A does not contain any non-reducing sugar. It only contains reducing because the mass did not increase. The rest, B, C, and D, they do contain non-reducing sugar because the mass increased from the original mass. But the difference will give me the mass of the non-reducing sugar, or will give me the mass of the copper oxide that will precipitate due to the presence of the non-reducing sugar. Now, 45 minus 30, what's the answer? Wake up with me. 45
B.
minus 30 is how much? 15 milligrams. So the 15 milligrams of copper oxide that appeared here in solution B was because of the non-reducing sugar. 55 minus 50 is 5. So here only 5 milligrams because of the non-reducing sugar. 75 minus 65 is 10 milligrams only because of non-reducing sugar. So the non-reducing sugar is found most in which solution, A, B, C, or D?
B.
B. Do you all understand? I'm going to do the Benedict's test, and this is going to give me a precipitate of copper 1 oxide, because we know that in the Benedict's, copper 2 sulfate will become reduced to give me copper 1 oxide, and this will be insoluble in water and will give me a red precipitate. When I boil with Benedict, let's say it will give me 30 milligrams of copper 1 oxide because reducing sugar is present. And then I want to see if I have non-reducing sugar or not. I will boil with HCl so that it will hydrolyze any non-reducing sugar. And then I'm going to neutralize it first before I carry out the Benedict's test again. I'll carry out the Benedict's test, and now it will give me a total of 45 milligrams of copper 1 oxide. 30, the first 30 was because of the reducing sugar. So the excess was because of the non-reducing sugar. Do you all understand how did we calculate this question? Any questions?
No.
Okay, good. Now, number 7. Where am I? Yes. Which polysaccharides contain 1 to 4 and 1 to 6 glycosidic bonds? You tell me. 1 to 6 glycosidic bonds means that this polysaccharide is branched. So let's summarize. Which polysaccharides are branched anyway? Who will tell me?
Amylopectin.
Very good. Amylopectin, and something else is branched.
Glycogen.
Glycogen. Very good. To cut a long story short, while you're studying, everything else is not branched. Cellulose is not branched. What else? We only took cellulose. Cellulose is not branched. Collagen is not branched. Hemoglobin is not branched. It is globular. The only molecules that can be described as branched is or are the amylopectin and the glycogen. So the answer is what? B.
B.
Okay. So this is a direct question, depends directly on how much you studied. Okay. Now let's move on. The diagram shows a small part of a polypeptide. What would the products be if the part shown was completely hydrolyzed? Okay, now this is not a very nice question also. Let's see how we will break this down. Obviously, this is an R group, and this is also an R group. So I'm going to look at an answer that has those two R groups. Let me see. The R group looks like this, and the R group looks like this. So where does the R group? I have this R group. Okay, this has the R group that we're looking for, and this we have the R group that we're looking for. Now let's look at the remaining amino acid. This amino acid, something is wrong with it. Do you realize what is wrong with it? There's a big mistake here in this amino acid. Can A be an amino acid?
No,
It
because
has
the
only
N
one
has
hydrogen.
one
Very
hydrogen.
good. You noticed that there has to be an H2, so it's not A. Okay, let's look at B. Also, I can see the R group here and the R group here.
Miss, the answer is D.
Okay, the answer is D. Wait, I want to see the answer is D. Let me see the difference between B and D. This is the R group, yes, that we're looking for, and this is the R group that we're looking for. And
Miss, B doesn't have the amino acid, the first one doesn't have an OH. It has a double bond with O and an H.
where?
The first amino acid, the C, the carboxyl. It has an O and an H, doesn't have an OH.
Oh, yes, and I was also looking for a mistake. Yes, there is no O. Yes, very good. Look at the functional groups. The functional group here is wrong. In A, the functional group here was wrong. So again, if you study the amino acids under functional groups, you will easily spot it. Now let's look at C and why is C wrong? Okay, again, it's easily spotted here. Look at C here, the carbon here, it has to be bonded to 4 bonds, not 3. So here it's a missing hydrogen. So also C is wrong. So the answer is D. What did we look for in this answer? We looked for the 2 functional groups and we found them. And the remaining amino acid was correct with the 2 functional groups. Did you all understand why it's D? Anyone has any questions? Actually, the question looks difficult, but it really turns out to be very easy. Anyone has any questions concerning this question? Okay, no questions, then I hope it's clear. Number 9, the enzyme trypsin hydrolyzes proteins to amino acids. Trypsin does not function when the pH is very low, as its 3D shape would be changed. What explains this change in 3D shape? Okay, when I have very low pH, then I have lots of hydrogen ions or little hydrogen ions.
Lots of hydrogen ions.
And hydrogen ions are positive or negative charged?
Positive.
Positive. Okay, now let's read. Hydrogen ions attach themselves to negatively charged R groups. Let's read B, okay? I will not ask you if it's correct or wrong. Hydrogen ions disrupt the disulfide bonds. Hydrogen ions increase hydrogen bonding between amino acids. Do hydrogen ions play any role in hydrogen bonding? Hydrogen ions reduce the affinity of hydrophilic R groups for water. Now, what is the most, by the way, this question, it depends a little bit on your critical thinking, logic. Hydrogen ions are positively charged. So they will bind to what?
Negatively charged R groups.
Very good. They will bind to R groups that are negatively charged. And we know that the R groups and their binding of the R groups, they determine the 3-dimensional shape of the enzyme. When these hydrogen ions, they bind to the R groups will not be able to form a precise 3-dimensional shape. So the answer should be A. Okay, let me see if the answer is A. 9, 8, yes, it's A. Okay, very good. 10. The flow chart shows some of the steps in the formation of collagen. I think we studied collagen so many times. We have 3 polypeptide chains held together by X bonds. Without moving on, what is X? These 3 polypeptide chains, what holds them together?
Peptide.
No.
R group interactions.
No. The 3.
All hydrogen.
The hydrogen bonds, it stabilizes them in place. They're not talking about bonding of 2 amino acids. If two amino acids are bonded together, I will say peptide. If we are talking about a precise three-dimensional shape, we will say the R groups will hold it in shape. We are talking about the triple helix of the collagen molecule. It's stabilized by hydrogen bonds. And then I have collagen, 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 collagen that are parallel. And the ends of these collagen molecules are bonded together by covalent crosslinks. Don't forget. So X held together by hydrogen bonds. They will form the triple helix. And then they are held together by intermolecular bonds, the covalent crosslinks. And then they form what? And then what? Many collagen molecules form what? And then finally form fibers. I'm trying to help you here. What is Y? Many collagen molecules will form
Fibrils.
fibrils. I don't know if you noticed, but this is similar to the cellulose structure. Cellulose, you have 1 to 4 glycosidic bonds holding the beta glucose molecules together, and they are parallel, straight chains, and they're also held by hydrogen bonds. Cellulose molecules will form microfibrils that will then form cellulose fibers. In collagen, we don't have anything called microfibrils. They're called fibrils. So which row correctly identifies X, Y, and Z? What is X? We said X is hydrogen, so the answer is either B or C. What is Y? Look here.
Fibrils.
Fibrils, not microfibrils. Microfibrils in cellulose. So the answer is definitely B. And let's look at Z, fibers. So they will confuse you. If you don't study well, you will get confused. So the answer for number 10 is B. Anyone has any questions? Any questions? Which
No.
molecules
No.
contain at least 2 double bonds?
A.
One minute.
D.
Saturated fatty acid. My dear, saturated fatty acid has 1 double bond only. The fatty acid here is saturated. So I have a chain of hydrocarbons, and then I have a carboxyl at one end, and a methyl group at the other end, which is CH3. This is 1 double bond. So 1 is not included. We want at least 2.
So D.
Yes, it's collagen and hemoglobin. Good. Oh, we finished. All correct? We have any mistakes?
No.
No, okay. Now, November 2020. So we finished June 2020, the 3 variants. Good. November, let's do this. A student carried out the Benedict's test on 2 different types of milk, X and Y. A sample of each type of milk was heated to 100 degrees in a water bath with Benedict solution, and the time taken for the first appearance of a color change was recorded. The results are shown in the table. So the type of milk, I have milk X and milk Y, and the type for first appearance of a color change with Benedict, 13 and 26. Now, logically, when it takes less time, then it will have more reducing sugar or less reducing sugar?
More.
More. So definitely, milk X has more reducing sugar. Which row shows the biological molecule the student detected in each sample of milk and the sample of milk with the highest concentration of this biological molecule? Okay, this is too easy. What's the answer? What are we detecting? Are we only detecting the presence of glucose or any reducing sugar?
Any
C.
reducing sugar.
Any reducing. Very good. And we said that the one that will have the first color change will have more reducing sugar. So the answer is A or C. Quickly, the answer is what?
C.
C. Okay, good. Let me see, who do I have? I swear those that don't attend are the biggest losers. Now here I have a diagram that shows the structure of a monomer. Now before I proceed, what is this monomer, Yassine? Look at carbon number 1.
An alpha glucose.
Good boy. Yes, it's an alpha glucose. Okay. Which molecule contains this monomer? Which contains this monomer? Amylopectin. Does it contain alpha glucose, Yasin? Amylopectin. I did not study it, which is part of starch. Amylose and amylopectin. Starch. What is the starch made of? Sucrose, glycogen. Do they all contain alpha glucose or not?
Yes.
Yes, they all do. So what's the answer?
B.
B. Very easy questions. You know, when are the questions difficult? When you don't study well. When you don't study well and you're easily confused, you will not get the questions correct. But when you study very, very well, you find them really easy, actually. Which molecules are formed by condensation, which involves a carboxyl group? Okay, I just want to ask a question. The carboxyl group, where is it found? I want to ask a question also. The carboxyl group, is it found in carbohydrates? Is it found in the glucose that we studied? Remember the glucose? Remember the straight chain? 1, 2,
No.
3. No, yes, 3, 4, 5, 6. We have two functional groups, carbonyl, COOH. It's called carbonyl, not carboxyl. I know it's confusing, but this is one carbonyl group. And at the other end, or carbon number 6, the hydroxymethyl, CH2OH. So there is actually no carboxyl group in any carbohydrate. The carboxyl group is COOH. Actually, the carboxyl group is only found in amino acids and in fatty acids. So it is only found in proteins and it is found in fats and triglycerides. So what's the answer? Anything that has a carbohydrate is not the answer.
D.
So what's the answer? What did you say?
D.
Yes, correct. A has amylopectin. No, it's not in any carbohydrate. B has amylopectin. No. C has amylopectin. No, actually the carbohydrate is the first option. So it actually makes it easier for you to exclude A, B, and C.
Miss,
Yes.
Miss, what is the difference again between the carboxyl group and the carbonyl group?
The carboxyl group, the carbonyl group and the carboxyl group. Wait, let me look at the pictures so that I get you a correct answer. The OH, not a double bond. Wait a minute. Where is the straight chain for the H, not OH? I wrote it incorrectly here. The carbonyl group is a double bond, O and H. This is a carboxyl, okay? But in a carbohydrate, there is no carboxyl. One functional group is the carbonyl. Carbonyl is C double bond O and H. The carboxyl is this. COOH. This is the carboxyl. OOH. So this is a carbonyl and this is a carboxyl. This is in a carbohydrate. I added the H by mistake because I'm used to it. So this is the carbonyl. Look here. Look here in the straight chain for the alpha glucose. This is the carbonyl group, C double bond OH. And this is the hydroxymethyl group at the other end. The COOH is only found in amino acids and fatty acids. Let me get you a picture of an amino acid. Where is the amino acid? This is an amino acid. This is a carboxyl group. Double bond O and then OH. The carbonyl, just remove the H. Very small differences that can confuse anyone. That's why you have to be very focused on the exam. Okay? Any questions?
No.
Okay, number 10. Which row correctly matches the function with the structural features of cellulose, collagen, glycogen, or triglyceride? Okay, now the molecule. Cellulose. Cellulose function, support? Yes. Molecules held together by hydrogen bonds. Is it held by hydrogen bonds? Yes. Branched chains. Is cellulose branched? No. Let's look at the triglyceride. Function, it's an energy source. Fibrous, a triglyceride is not fibrous, of course. Molecules held together by hydrogen bonds, no. By ester, so no. Branch chains, no. Okay, do I move on? What's the answer for 10?
A.
A. From the first option, everything is correct. Do you want me to move on so that we check that the rest are wrong? Let's take C, for example, collagen. Function, strengthening. Okay, fibrous, collagen. Is it fibrous? Yes, it is fibrous. Molecules held together by hydrogen bonds? Yes. Branched. Is collagen branched?
No.
No, collagen is not branched. To cut a long story short so that you're never confused, the only branching molecules, the amylopectin and the glycogen. So C is wrong and you'll find a mistake in each and every one of them. Okay, let's finish this exam. This question is not nice at all. The diagram shows the amino acids in a polypeptide. An enzyme catalyzes the hydrolysis of any peptide bond between a non-polar amino acid and any polar amino acid. How many small peptides and single amino acids will be formed by the action of this enzyme? Okay. Now, they said here, they only gave you a hint and they told you that, that the enzyme will catalyze any peptide bond between a non-polar amino acid and any polar amino acid. You will go to the key and you will find that the non-polar amino acid is oval and the remaining amino acids are polar. So what would you do to see where the enzyme will catalyze? You will go look for which amino acid, the non-polar or the polar?
Non-polar.
The non-polar. You will go to the oval-shaped amino acids and you will check if they are bonded to a polar amino acid and you will make a break. So let's do it together. I'm going to start from the base here and then I'm going to find here one oval amino acid. One oval amino acid, I'm going to make a break because this is non-polar and this is polar. And there's another non-polar amino acid, but I will not make any breaks here because this is non-polar and non-polar. They said non-polar and polar. So this will be a break here. Are you with me? Do you know what I'm doing?
Yes.
Okay, I'll stop here. This is non-polar. So there is a break here. Okay, this is non-polar. So there is a break here between the non-polar and the polar. I'll move on, move on, move on, move on, move on, move on. I'll stop here because this is non-polar. And there will be a break here. I'll catalyze. And also there is a break here because this is non-polar and this is polar. And we'll move on, move on, move on, move on. I'll find a non-polar amino acid here. So there has to be a break here. Not between two adjacent non-polars, but one, but a break between a non-polar and a polar. So there has to be a break here. Okay. I'll move on. All these are polar, 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 polar. I'll stop here. Non-polar. So there will be a break here. And then again, polar and non-polar. I'll break this here. And then I'll move on. All these are polar, 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 polar. I'll stop here. There is non-polar and polar. And also non-polar and polar. I hope you're with me. Okay, I always stop at any oval amino acid because this will be a non-polar amino acid. And if next to it I have a polar amino acid, I'll make a break. And then all these are non-polar. There has to be a break here. And a break here. And then I will count. You have here the two options of the number of small peptides and the number of single amino acids. Let's count how many single amino acids did we create. This is 1. This is 2. There should be 1 here. 3. And this is a single amino acid, 4. So 4 is the only number here. So the answer has to be D. To confirm, we will count the number of small polypeptides. They should be 13. 1, 2, 3. This is 4. 5. 6, 7, 8, 9, 10, 11, 12, 13. Do you understand what we did here in this question? Do you understand what we did it and how we did it?
Miss, the only thing I don't understand is the breaking.
They said here that the breaking happens between non-polar and polar. Those 3 shapes are polar. The only shape that is different is the oval. So I will stop at every oval shape and I will make a break. As long as this oval shape is next to any one of the polar amino acids, then there will be a break. This is what I did. I just moved and I stopped at every oval. Let's say this here is the histamine and the valine. This valine is nonpolar. Next to it is a nonpolar, so there's a break here. The valine and the PHE, they're both nonpolar. There's no break between them because they said that the enzyme will break between the non-polar and polar. So it will be between the PHE and the THR. I'll make a break here. Better, Mariam?
Yes, I got it.
Okay. This question is not very nice, actually, but we have to solve it. Which properties of water reduce temperature changes inside cells? Cohesion or latent heat of vaporization or specific heat capacity?
C.
Yes. Cohesion helps in the movement of the molecules, of the water molecules up an xylem column, for example. Latent heat of vaporization is the amount of heat needed for the water to evaporate, so it will cool you down. But specific heat capacity is the amount of heat needed to raise the temperature of 1 kilogram of water by 1 degree. And this is very high. So that is why water is stable most of the time and it doesn't change, it doesn't fluctuate in temperature. So the answer is D. Okay, these are the properties of water. Did we do any mistakes? Did you do any mistakes? No mistakes?
No.
Not yet?
No mistake.
Type, we solve another exam. Let's solve one final exam and then I'll let you go. Which pair of monosaccharides forms sucrose? Now, very easy. I will not even... Yalla. C or D? Because sucrose is alpha glucose plus fructose.
C,
Yes, C. Why did you choose C?
because it's alpha glucose.
Yes, very, very, very easy. Okay, good. The diagram shows the structures of 2 amino acids. These amino acids will form part of a protein that has a tertiary structure. Which numbered groups could form hydrogen bonds to maintain the tertiary structure of the protein? Okay, here we're talking about tertiary structure. So these bonds that will keep or maintain the precise 3-dimensional shape happen between which groups?
D,
Yes, you are correct, D. Why did you choose D? Because the tertiary structure is held in place by the bonds between the R groups. Okay? It can never be 1 or 2 or 4 or 5 because these are not the R groups. Clear? Any questions?
Yes.
Good. 10.
No.
Mariam, you have a question, my dear?
No.
Okay. 10. Mariam, answer number 10. Which row about the structure of a protein is correct? Let me minimize that. So A, B, C, and D. Read it and tell me. Primary structure. Look for one correct answer, and then most probably it will be the right one. The primary structure, A, B, C, or D? Number of amino acids. The order of the amino acids encoded by DNA. Now, the primary structure, we always use a certain word. Huh? Yalla.
sequence. A sequence of amino acids
Sequence.
فدي.
Very good. Sequence. Correct. And to make sure tertiary structure formed as a result of interaction of the side chains. Yes. And the quaternary structure formed by the linking together of more than one polypeptide. Yes, it has to be D. Okay. 11.
Miss, why it's not the order of amino acid?
Let me see. It's the order of the amino acids present in a protein encoded by DNA. Let's look at the remaining tertiary structure. Is the shape formed by folding of a polypeptide and held together by hydrogen bonds. This is wrong. The tertiary structure is not held together by hydrogen bonds only. Hydrogen and hydrophobic and ionic and disulfide. Quaternary structure contains two types of polypeptide that interact, forming the shape of the protein. Only two. Quaternary structure can be any number of polypeptides. The three of them have to be correct. This maybe it's not wrong, but the remaining are not right. And these are more correct. They are perfect. D is the perfect answer. Primary structure is the sequence of amino acids. Tertiary structure is formed as a result of the interaction of the side chains, not only hydrogen bonds. Quaternary structure is formed by the linking together of more than one polypeptide. Two or more, not only two. So B is not right. D is more correct. Okay, good. Now we have number 11. Which row correctly identifies structural features of a collagen molecule and a hemoglobin molecule? I think this is the last question. Now, molecule consists of more than one polypeptide, and which is globular? This is so easy. What's the answer?
B.
B. Globular is hemoglobin. And both of them are more than one polypeptide working together. Okay, very good. Another exam? Oh, let's solve this exam so that we finish June and November variants. Okay. A student carried out tests for biological molecules on the same sample of milk. The tests and the results were as follows. Heating to 80 degrees with Benedict gave a red color. So the milk contains reducing sugar or not? When it gives you a red color?
It does have reducing sugar.
Yes. And then we boil with acid, followed by neutralization, and then we heat again to 80 degrees with Benedict. Solution also gave a red color. They did not say a darker color. I want to just put this in consideration. Adding burette gave a purple color. So it contains protein or no protein? We said it has reducing sugar. Huh?
Protein.
Yes, there is protein. Adding iodine will give a yellow color. So there is starch or no starch?
No starch.
No starch, no starch. Which biological molecules must be present in the milk? Must, and the must is in bold. So non-reducing sugars, do they have to be present?
No.
No, because when I boiled with acid, followed by neutralization, and I heated with Benedict, it also gave me a red color. So this means that maybe it is present, but definitely reducing sugars are present. But non-reducing sugars, I'm not sure. So one is not necessary. So I will exclude A and B. And then I have C and D. What are the answers? Which answer do you think? 2 and 3. Protein and or 3 and 4. No, it is C. Because we said that there is no starch. Remember?
Yes.
Okay. Now, which row is correct for carbohydrates? What's a monomer? A polymer and a macromolecule. Don't forget that a polymer is a macromolecule also. Monomer, this is a monomer, this is a monomer, this is a monomer, this is a monomer. Okay, so the monomer will not help you. Polymer. Amylose.
Yes, sucrose isn't a monomer.
Excuse me?
Sucrose is not a monomer.
Oh, شطورة. Yes, of course it's not a monomer, so we will exclude D. So now we have A, B, and C. Now polymer. Amylose is a polymer. Deoxyribose is not a polymer. Deoxyribose is actually a monosaccharide. We will study it when we study ATP. And we will study it when we study DNA. So it's not B. So now we have A and C available. Now, is it A or C? Macromolecule. Is deoxyribose a macromolecule?
No.
No. The deoxyribose is found in the DNA. It's the sugar that is found in the DNA structure. We still did not take DNA. It's a monosaccharide, actually. So the answer is A. 9. Which molecules contain 1 to 6 glycosidic bonds? In other words, which molecules are branched?
B.
Why? What about starch? Starch is also part of it, amylose and amylopectin. Amylopectin is branched. So it's A.
Oh, okay. I thought they were talking about. Okay, خلاص.
It's okay. We all get confused. We all get confused. And sometimes you get confused because you read the question very quickly. Okay? It's not that you don't know the answer. It's because you read it quickly and sometimes you bypass it. Which row correctly shows the ratio of carbon atoms to hydrogen atoms for glucose, a saturated fatty acid, and sucrose? Now, the ratio of carbon atoms to hydrogen atoms. Okay. Now, the glucose will be easy. C6H12O6. They want the ratio of carbon to hydrogen.
1 to 2.
C6H12O6. Of course, 1 to 2. So the answer is either A or B. Now, sucrose. I advise you to ignore the saturated fatty acid for now and go to sucrose. What is
It would be A.
A.
It's A, yes, because we said that a disaccharide, it will be C12H, what? 22,
22.
and O how much?
11.
22 divided by 12, we'll find it is 1.8. So the ratio will be 1 to 1.8. So the answer will be A. And also saturated fatty acids, which is the chain of hydrocarbons, and it is saturated, so there is no double bond. So every carbon is bonded to 2 hydrogen. So the ratio will also be 1 to 2, 1 carbon to 2 hydrogen. Yes? So the answer is A. You all agree? It's a tricky question, a little bit tricky. 11. Which row shows a correct comparison between a phospholipid molecule and a triglyceride molecule? Now, this I think is very easy. Who will give me an answer? Phospholipid and what did you say?
C.
Fuel ester bond. Of course. Contain a hydrophobic phosphate group phospholipid. It's not hydrophobic, it's hydrophilic. Contains 1 glycerol molecule. Contains 3. No, wrong. Okay, yes, C. More fun. Yes, it's C. 12. Adult human hemoglobin typically consists of 2 alpha chains and 2 beta chains. Approximately 5% of humans have 1 amino acid in the beta chain that has been changed. This is the mutation that we talked about when we said we have GAG. This A will be mutated to GTG, okay? And what happens is that 1 polar amino acid, the glutamic acid, will be converted or will be changed into 1 non-polar amino acid, the valine amino acid. This mutation, we'll study it again in protein synthesis. Okay, this change affects the structure and stability of hemoglobin. Which levels of protein structure could be affected in the hemoglobin of the humans with the changed amino acid? This is also very easy. When we change 1 base, and then we will change 1 amino acid, and then we will change the, what's the answer? D.
D.
Change everything. What did you say? Not.
D.
If we change the primary structure, everything else will be affected. We will change the sequence. Therefore, we will change the amino acids made. We will change their R groups. We will change their folding. We will change their R groups and their tertiary folding and their quaternary folding. Everything is affected. Okay? And we'll study this more when we study mutation. Oh, we finished. Did we do any mistakes?
No.
Really? Okay, very good. This is 2021. So we finished June 2020 and November 2020, the 3 variants. So we did 6 exams. That's a very good achievement. Okay, those who did not attend, they are the losers. Fine. You have any questions? This enough, or we solve some more? Or you will solve alone?
We will solve it.
Okay.
Alone.
Any questions before I go? When
No,
is
don't.
the quiz, Mariam? We said that you have a quiz. When is the quiz?
4 December.
4th of December. When is the 4th of December?
On Monday.
That's next Monday? No, we'll make it next Wednesday. Give me a chance to print it. I'll announce. I'll write on Google Classroom and I'll write on the WhatsApp group. Serene, you will attend next week?
No.
Why not? You're still not good?
لسه أيوة هقعد بيها كمان أسبوع.
You cannot come to school with it?
Unfortunately not.
Okay. I actually recorded one of the sessions. I'll send it to you.
Okay.
One of the
Thanks
classes.
a lot.
You're welcome, my dear. Okay, bye.
Bye.